For our first laboratory exercise, we focus on external morphology of invertebrates, especially insects and arachnids. Entomology is the study of insects and related arthropods. These organisms have segmented bodies, jointed legs, and hard exoskeletons, which make them successful and diverse in many environments. The purpose of this exercise was to identify and describe the external body parts of insects and arachnids, like the antenna, mouth parts, eyes, legs, wings, and ovipositor, and to understand how these structures help them function and survive. We observed that insects have three body regions, the head, thorax, and abdomen, with three pairs of legs and usually wings, while arachnids have two regions, the cephalothorax and abdomen, with four pairs of legs with no antenna or wings. This activity helps us become more familiar with their function forms and adaptations.
Did you know the phylum Arthropoda accounts for nearly 55% of known species on Earth? While they are vital for pollination and recycling nutrients, some of these are pests and may cause economic losses and diseases. The purpose of the second laboratory exercise explores the needed instruments for collecting arthropods, as knowing how to handle insects is essential in studying biodiversity and assessing pests. For simple above-the-surface collection, you would need bond paper that will serve as a platform for bait, which is a mixture of tuna for protein and the peanut butter for sugar. The insects are then stunned with an alcohol sprayer and stored in plastic bags. Minute specimens are collected using a moistened fine paintbrush. For flying specimens, a net is used, and the captured insects will be placed in a kill jar, a wide mouth container with cotton balls soaked with chloroform, and a cardboard with holes.
For any further development in understanding animals, genetic, morphological, or ecological, it is important to note that prior collection is everything. Why? Because proper collection is the first step to ensuring correct representation of a biological specimen for scientific studies. In entomology, collection happens in two particular ways, active and passive. Laboratory 3 explores these two cases of collection where active collection is the exertion of physical effort, think sweep netting or hand collecting a specimen, which the researcher directly captures the insect. And passive collection, the setup, where a student deploys a tool like a pitfall trap or a food bait and let the insect's natural behavior do the work for their capture. Here are the pictures and documentation for collected specimens from baits during this laboratory exercise.
In biology, organisms are classified based on their similarities and evolutionary relationships, from broad groups like domains to specific ones like species. Among these groups, one of the most diverse and fascinating is the arthropods, a group that includes insects, spiders, and crustaceans. Arthropods share unique features such as segmented bodies for movement, jointed legs for crawling, swimming, or flying, and tough exoskeleton for protection. As invertebrates, they lack a backbone and have an open circulatory system where blood flows freely, along with bilateral symmetry, meaning both sides of their bodies mirror each other.
Now I am going to talk about the different arthropod orders and their unique features. Firstly, the Diptera are true flies, which are unique because they only have one functional pair of wings, while the second pair form small knobs called halteres for balance. Next is the Coleoptera or the beetles, which have hard forewings called elytra that protect their hindwings. Next is for the Lepidoptera, which includes butterflies and moths, which have scaled wings and a coiled proboscis for feeding on nectar. For Orthopteras, like grasshoppers, their jumping legs are their most distinct feature. For Hymenopteras, which include ants, they have elbowed antennae and undergo complete metamorphosis. While for Siphonopteras, or fleas, they are wingless with piercing, sucking mouthparts and flattened bodies which are adapted for parasitic life. For Hemiptera or true bugs, they are characterized by their hemelytra forewings that are partly leather and partly membranous. Lastly, for the Aphidoidea or aphids, they are distinct because they can give birth without mating through viviparity and parthenogenesis.
In this activity, we identified three Hymenoptera or ant species collected from our campus. All belong to the Formicidae family known for their elbowed antennae, forward-facing mouthparts, and complete metamorphosis. Specimen 1 or Solenopsis invicta or the fire ant is reddish-brown with darker gaster. Specimen 2 or Tetramorium immigrans is dark brown to black and covered in fine woolly hairs. And specimen 3, Cardiocondyle emery, is lighter, ranging from brown to yellowish tones. And though they differ in appearance, all share the key traits of ants, a perfect example of diversity within the Hymenoptera order.
Proper insect mounting is essential for accurate identification and preservation. To begin, an entomological puncture is used to make paper tips, which should be in a concave position and placed between the first and second pair of legs. These tips are inserted into an entomological pins using a pinning block to ensure desired height. Once prepared, these pin tips are placed into an eguchi ball, which holds them securely and allows the specimen to be observed from different anatomical perspectives. Before placement, an entomological probe is used to gently spread the insect's legs. The same probe can also be used to put a small amount of water-based glue to the paper tip and to carefully position the specimen. Once mounted, labels can be put below the specimen to provide essential taxonomic information. Finally, a dissecting microscope can be used to observe the ant's morphological traits.
This laboratory exercise was a collaborative effort among 6 groups. Together, we explored the morphological characters of arthropods, focusing on ants under the family Formicidae. We learned that morphology, the study of form and structure, is essential for identifying species. Each group worked on labeling ant body parts such as the head, thorax, and abdomen, as well as specific features like the antenna, clypeus, carina, and etc., which are important in building dichotomy schemes for classification.
So this collaborative activity helped us appreciate how detailed entomology can be. Even the smallest structures like ridges or fine hairs can distinguish one species from another. By carefully observing and labeling these parts, we develop a deeper understanding of insect anatomy and identification. Overall, this laboratory work strengthened our teamwork, sharpened our observation skills, and prepared us for future entomological studies involving arthropod classification and morphology based identification.
Studying arthropods and their external structure help us understand their adaptation, functions, and ecological roles. Through proper taxonomy, identification, and collection methods, we can accurately classify species and document their diversity. These practices are essential for recognizing the importance of arthropods in nutrient cycling, pollination, and maintaining ecological balance, showing how vital they are in sustaining life on Earth. This has been Group 3, and thank you for watching.